Hey everyone, it's your boy Mr. Jennings here. Welcome to Big Orange Biology. Today we're going to talk to you about photosynthesis and wrap up Unit 3 Energetics. Now, photosynthesis is making life from light and air. It's a very valuable process for plants all around the globe and some photosynthetic bacteria. Now, what does it mean to be a plant? Considering the majority of the organisms that do photosynthesis are plant-based, plants need to be able to do the following things. One, they need to be able to collect light energy. They got to be able to transform that light energy into chemical energy. Now, that light energy needs to be able to be stored, hence the need for chemical energy. Now, it has to be stable. So ATP is not very stable. ATP is a very unstable molecule. That means we need to make it something like glucose. Next, we need to be able to build various things. So here's the thing. Photosynthesis is the only way plants obtain their nutrients. So, or at least harness their nutrients. And they need to get building block atoms from the environment, like CO2, water, and the trace elements like nitrogen, potassium, and phosphorus. So they need all these to produce all the organic molecules they need for life. They need to be able to make carbs, proteins, lipids, and nucleic acids using photosynthesis. So there's a lot having to do with one chemical reaction. Now that relates to us because we eat the plants, and that's how we get a lot of our nutrients ourselves. Now the plant structure helps this out. So remember, structure equals function. The ability to obtain raw materials such as sunlight comes from things like the leaves. They're great for being solar collectors, like big solar panels for plants. CO2. The leaves also got you there. Underneath the leaf, you're going to find these things called stomates. Stomates help the gas exchange. They're going to allow CO2 to come in. Also get rid of that pesky oxygen. That's going to come out later. Water. Now we need water too. Water is going to be great to come up with the roots. That's going to use osmotic pressure to do that. And the nutrients. Guess what? We need nutrients, but you know where we can find a lot of the nutrients? The soil. Guess what helps with the soil? The roots. So all those trace nutrients, they can get taken up by the roots via the same processes with water. Now the chloroplast is the key organelle here. It is found in the leaves. Here you go, a little cross-section of the leaf. If you look at any kind of cell in the leaf, you're going to find a ton of these little organelles known as the chloroplast. Now, chloroplast is a small photosynthetic organelle in plants. The big thing is it has highly evolved from bacteria. It's more likely an endosymbiote. And we're going to learn a little bit more about it. Now, the chloroplast does contain the pigment known as chlorophyll. Now, chlorophyll's big thing is it allows the sun and CO2 to come together to make energy and sugar. Now, photosynthesis is two series of reactions in one. First off, you have the light reactions. These are known as the light-dependent reactions. They require light in order to function. And they are energy conversion processes. They're going to take that solar energy and make something chemical out of it. Typically, they're going to make ATP and NADPH. NADPH is an electron carrier, much like its similar cousins NADH and FADH2 from the Krebs cycle and other cellular respiration processes. Now, the Calvin cycle is a process. It's called the light-independent reaction, and it builds sugars typically. But it can also build other things. It can use chemical energy found from ATP and NADPH to help reduce CO2 and to help synthesize C6H12O6. Now, the light reactions utilize an electron transport chain in order to convert that light energy into chemical energy. Much like any other process, we're trying to make ATP, so one of the best ways to make ATP is using ATP synthase, which is usually at the end of a ETC. Now, proteins found in the organelle membrane are going to help carry this out. So, electron acceptors like NADPH are going to be formed here, unlike in cellular respiration where they're consumed, or at least recycled. Now, like any electron transport chain, every time I move an electron across one of these proteins, it's going to allow the movement of a proton across the gradient. And in doing so, I want to help build up a high concentration of hydrogen ions, which means they need to go somewhere, so they're going to go right back through ATP synthase to make a bunch of ATP. Awesome. And this just modeled that for you. So the electron transport chain of photosynthesis is very similar, but it is slightly different. We'll elaborate more on this in class. But essentially, chlorophyll is going to help transform that light energy into the chemical energy of ATP. They use electron carriers like NADPH and the intermediates like ATP. It's going to help produce oxygen, unlike the other one. And in doing so, it's going to be a very, very valuable process. Now, this is all helped out because of the pigments of photosynthesis. See, photosynthesis utilizes pigments like chlorophyll and other things such as carotenoids and xanthanoids. Now, they're embedded in the thylakoid membrane. They're going to be called photosystems, and whenever light hits them, they're going to get really excited and lose electrons. Now, photophosphorylation is the process of which you use light to add phosphates. Both of these reactions are going to be dealing with phosphorylation, at least to some extent. We're going to make ATP, so we're going to need to be able to do it with light. Now, typically, there's two forms of photophosphorylation. There's non-cyclic, which is what it sounds like. It's a linear. Photosynthesis 2 gets excited, moves electrons around. Photosystem 1 moves it around to make NADPH. Pretty easy. Also, it's going to make a little bit of ATP. Pretty easy. Now, cyclic processes come along, and that's basically when you have a bunch of NADPH, but not enough ATP. So photosystem 2 is going to get excited, goes to photosystem 1, but instead of going down to making NADPH, it just makes more ATP. It kind of skips the NADPH part. And in doing so, it's going to make more ATP and keep doing so until enough ATP has been made. Now, why do they do this? Well, CO2, the raw ingredient along with water, has very little chemical energy. CO2 is abundant, and even more abundant these days. But CO2 has very little chemical energy. It's very, very oxidized. It's fully oxidized. It's a 1 to 2 ratio of oxygen to carbon. Sorry, carbon to oxygen. Not a lot of room for growth. Now, glucose, on the other hand, now that's a molecule. That's got that 1 to 1 ratio. That means it's highly reduced. There's room for oxidation. So this is going to be utilizing a synthesis reaction, which is a anabolic process, which is endergonic. Now, to make something, when endergonic's around, you got to remember, that needs a lot of energy to work. So the reduction of CO2 to glucose takes a lot of time, and it takes many pre-steps. Okay, it doesn't take a lot, but it takes a lot of work. It takes a lot of energy. So you're going to have a lot of small steps to kind of help that out. And each is going to be helped out by a specific enzyme. Not only is it going to use a specific enzyme, it's going to use the energy stored in these ATP and NADPH to help get around this activation barrier that endergonic processes have. So reducing CO2 to glucose takes a lot of energy. Some enzymes to help out. ATP and NADPH really help provide that energy. So we need those from the light reaction. So light reaction is necessary in order to build up the energy to overcome the cost of activation for the reaction. Now, one of those key enzymes, and mainly the super important enzyme you need to know, is the enzyme rubisco. Now, rubisco is highly involved in the Calvin cycle. It is the enzyme that allows us to fix carbon from the air. It takes CO2 and slaps it onto RUBP, which allows it to enter into the process. Allows it to go into the whole Calvin cycle itself. It's the single most important enzyme in the world. It is also the most abundant enzyme in the world because every plant has at least hundreds of thousands of copies. Now, photosynthesis is two reactions. Got light reactions making ATP and NADPH. It's going to make some water. It's going to take up water, and it's going to make some oxygen. Calvin cycle, on the other hand, it's going to take the CO2. It's going to consume it. It's going to produce a special sugar called G3P. More on that in class. And it's going to regenerate ADP, so it's going to consume ATP. And it's going to regenerate NADP, which means it consumes NADPH in the process. Now, one of the problems plants face is controlling water loss. See, plants have some adaptation issues. They're not like animals that can just get removed. Plants are largely stuck where they're at. So controlling water loss from leaves is very hard. Hot and dry days, here's the thing. Stomates have to close in order to conserve water. So water loss is a serious issue, so they're going to close up. And that's because the guard cells, the little gate channels on the stomate there, the gated channels, the problem is water controls it. So we have a lot of water, the stomate's open using turgor pressure. A lot of less water, stomate's closed because, again, turgor pressure. It's an adaptation of living on land. It helps, water loss, but it creates a lot of problems. Now, when stomates close up here, you're going to have some issues. Closed stomates lead to a problem. It kind of shuts off some things. O2 builds up, so oxygen is going to start building up from the light reactions. Now, CO2 is going to be consumed and depleted, so the CO2 stores are going to be going away from the Calvin cycle. This is why the problem is for the Calvin cycle. See, not enough CO2 and too much oxygen causes a problem for our boy Rubisco here. Rubisco is the carbon fixation enzyme. CO2 is the optimal substrate, however, when oxygen is high, Rubisco will bond with oxygen. It's a competitive substrate, it's a competitive inhibitor. See, high amounts of oxygen inhibits the Calvin cycle. It's a negative feedback. And in doing so, helps reduce the consumption of CO2, and that's a problem. It leads to the oxidation of RUBP instead of the reduction, and it causes a breakdown of sugars. So by putting too much oxygen, Rubisco is going to become the very thing it's sworn not to be, and break down the sugars instead of building them. And that's called photorespiration. That's a big issue for a lot of plants. See, when CO2 is abundant, the Calvin cycle flows like normal. Hey, it looks great. These are called C3 plants because they make a 3-carbon sugar. Great. When O2 is too high, Rubisco is just like, hey, I'm going to go on and do something else. It goes on to the mitochondria, and it's going to be lost as CO2 without making any ATP. So oxygen is just going to get CO2, oxygen's too much there, bonds to Rubisco, and Rubisco is like, okay, I'm going to go take the sugar over to cellular respiration and make it happen. Photorespiration. It's a very serious issue for plants. See, it causes about 50% deficiency. Now, what it does is it reduces the production of photosynthesis, meaning no ATP is produced, no glucose is produced. And if you were to reduce the amount of photorespiration, well, they'll become 50% more efficient. There's a strong selective pressure, though, for plants to evolve in pathways. It's not good to have, C3 pathways because you have the problem of being stuck in photorespiration. So reducing photorespiration is a very, very strong advantage. So some plants have separated the carbon cycle from the Calvin cycle. These are known as C4 plants. They physically keep the Calvin cycle away from away from light reaction. So different cells to fix carbons versus where Calvin cycle occurs, stores carbon in what's called a 4-carbon compound, makes what's called BEP carboxyls. It also has a totally different leaf structure. Another type of plant is called the CAN plant. Now they separate by carbon fixation from the Calvin cycle by time of day. Basically, they fix carbon at night when loss is not an issue, and they do Calvin cycle during the day to help continue it out. So the CO2 is being fixed. It's stored as a 4-carbon compound known as Crushelian acid, and it's going to be used later during the day in the plant. And those are you typically C4 plants. Again, they capture CO2. They keep things a little separate. It's a great adaptation to dry, hot climates. Your monocots, they do this a lot. Things like your grasses, sugarcane, corn. They keep their stomata closed a lot because it's so hot. They have a different leaf anatomy to kind of help make sure photorespiration occurs. That's this anatomy here. They physically keep this. Just goes right through it. Now, Crushelian acid metabolism in plants is for hot, dry climates. Separate carbon fixation by day. Stomates stay closed during the day, open at night. At night, they open up, allows carbon fixation to occur, and stores them as Crushelian acid. In the day, it was the CO2 into the CO2 from the 4 carbon acids that are breaking them down and release those carbons back into the Calvin cycle. This allows the increased concentration of CO2 in the cells, and this is found in succulents like aloe vera, some cacti, and your pineapples. This solves the CO2 oxygen gas exchange versus water loss challenge in 2 different novel ways. See, C3's got a problem though, it's evolutionary baggage. See, Rubisco evolved in high carbon dioxide atmospheres. Think, the atmosphere during the Jurassic period, full of carbon. There was no strong selection against it. So Rubisco got played both sides of the field. Nowadays, the atmosphere is a little different. 21% of the atmosphere is oxygen, 0.3% is CO2. Photorespiration can drain away about 50% of that carbon fixation on a very hot, dry day during, say, a drought. So strong selective pressure helps kind of keep things better, find better ways. Overall, though, the process, it directs towards fitness. Okay, let's wrap things up here. So, photorespiration is a critical process. It is Photosynthesis is a critical process. A lot of things happen. It's used to make sugars and all sorts of very important macromolecules in addition to those. Now, the overall process is something we'll discuss further in class, but overall, plants have been doing this for a very long time, and they've adapted new ways to become more fit to their environment. Overall, though, it's really critical to understand the general pathways and the trends. That said, I hope you had a great day. This wraps things up for this unit. Next unit we're coming in is cell communication. So we're going to get started talking about mitosis here soon. But thank you for watching. Have a great day. Bye.